Good morning, good afternoon, good evening, participants from all over the world. Today we're going to talk about sexual plant reproduction, which is an amazing and still largely very mysterious process in plant sciences. Imagine a single cell, the pollen grain, capable of finding an egg cell with an amazing speed, and it can travel along a very large distance, up to 20 or 30 centimeters. And then it is capable of finding that other single cell, the egg cell, which is carefully hidden in the ovules, which are carefully hidden in the carpels. And the other process is also very interesting and mysterious. The fertilization of this single egg cell in a double fertilization event. And then the zygote is developing into a seed. And the seed is, of course, the heart and the starting point of all of our crops. There's also a third process, which is of very much interest, which is occurring in several plant species, which is the fertilization of the development of a single egg cell into a seed without fertilization. And that is the process of apomixis, and that is the topic of today. How does that work? How can we apply the process of apomixis once we understand it? And to address that, we have gathered speakers from all over the world, from the Netherlands, from Italy, from Canada, and from China. We also have a panel who is going to discuss the working mechanism of apomixis and also the application of apomixis. But before we go into the panel, I would like to introduce you with the first speaker of today, Professor Lucia Colombo. She has obtained her doctoral degree in molecular and cellular biology in 1993. From 1993 until 1997, Lucia and we worked together when she worked as a postdoc in Wageningen University and Plant Research International. In 1999, she got a permanent position at the University of Milan. Lucia has been one of the pioneers in the study of plant development in Italy, where she obtained important recognition for her research activities. In 2003, she was selected for a prize of the President of the Italian Republic, Mr. Ciampi, for her outstanding work. She became full professor in general botany in 2010, and in 2018, the Biosciences Department has been selected, where she is the head of, as one of the Italian Department of Excellences in Biological Sciences. Since several years, the focus of the research of Lucia's team has been the development of reproductive structures in angiosperms. So Lucia, I very much welcome you today, and the title of Lucia's presentation is Reproduction of Plants, an exciting field of research and opportunities. The floor is yours, Lucia.
Well, thank you very much. Thanks for the nice introduction, and good afternoon to all of you. So I want first to thank Arjen and all the KeyGene staff for the organization of this interesting and timing workshop on apomixis. I've been asked and invited to give an overview on the research opportunity in the field of plant reproduction. This invitation is a huge risk for all of you because I can talk for hours about plant reproduction. However, because this, as Arjen said, is a topic that fascinated me since I was a PhD student. However, don't worry, I will limit my talk within the 12-14 minutes assigned to me. So, as all of you knows, plant reproduction stands at the basis of food production and also production of many not food products. And is an extremely complex and multi-step process that in flowering plant required the formation of flowers that contain male and female reproductive organs, named stamen and pistil. So the male gametes are formed inside the pollen that itself develop in the stamen, whereas in the pistil, the female gametes are formed inside the ovules. So if we want to summarize plant reproduction, we can divide it in three following major phases. The first one, of course, is flowering, the switch from vegetative to reproductive phase. Once that we have the switch, the next step is flower formation and fertilization, upon which we have the third phase in which seed and fruit develop. As I said before, I could talk for hours on the achievement obtained in this field. And I can also make a very, very long list on open questions that still have to be addressed. Therefore, I had to make a selection, and I decided to focus on some examples based on the research performed in the Department of Bioscience at the University of Milan, that hosts many plant scientists working on plant reproduction, including myself. So the first step is flowering, as I said, and is the switch between vegetative phase and the reproductive phase. This switch is controlled by an extremely complex genetic network interacting with environmental clues, as for instance, temperature, vernalization, photoperiod. Here is an example. So rice is a plant, is a typical tropical plant, which normally flower under short day condition, as you can see in this slide. However, in Italy, one of the most northern area where they grow rice, the plant flower under long day condition, because our variety, so the Italian variety, are insensitive to day length. Of course, we are very much interested in the genetic base of the insensitive to the photoperiod, because this will allow us to use in the future tropical rice variety in our breeding program. Of course, once that the flower form, fertilization can take place. And to make a long story short, I summarize in this slide what is going on during fertilization. So the two male gametes are brought to the pistil, the female reproductive organ, through the pollen. And through the pollen tube, the two gametes are delivered inside the ovules. And we have a double fertilization. One male gamete fertilizes the egg cell to give origin to the embryo. And the second one fertilizes the central cell to give origin to the endosperm, the feeding tissue of the embryo. So upon double fertilization, we have that ovules develop into seed and pistil are developed into fruit. So this process is extremely important and is consisting in many steps. Of course, in Milano, there are several groups working and try to study the genetic base or different phase of fertilization. And some of these groups are actually interested in identifying genes that could make less sensitive this process to heat waves that often compromise the entire process. So heat waves is becoming more and more a problem because it's becoming more and more frequently, especially in Italy, but not only in Italy. So in Milano, we are also very much interested in making a strategy to increase seed numbers. So seed number for plant. So we have developed two different strategies depending on inflorescence architectures. One of these strategies is used to increase seed number for flowers in which for plant in which pistil contain multiple ovules, for instance, like Brassicaceae. Of course, more ovules means also more seeds. Another type of strategies that you see on the right side of this slide is to increase flower number for plant. And also in this case, more flower means more female reproductive organs, more seeds. So this is, of course, a very interesting and important issue because will allow to produce more seeds without increasing the amount of lands required to get this increase in seed production. And this is one of the targets in sustainable agriculture. But it's just not a matter of seed number, but also of seed quality. So another challenging topic is to try to implement seed side and quality. And there are a few groups that are also investigating these topics. Here there are some pictures in which clearly see how seed could be, of course, be formed with increase and quality characteristic in both balanced and unbalanced crosses. And when I talk about seed quality, I mean also germination efficiency. So all these examples that I brought to your attention, they are mainly coming from sexual reproduction. So you have to keep in mind that 90% of the angiosperm have hermaphrodite flowers. So in principle, they can self-fertilize themselves. However, many species have developed self-incompatibility mechanisms to promote allogamy. So therefore, fertilization has to occur between gametes produced by plants of the same species, but genetically diverse. Fertilization in this way in nature guarantees genetic variability, but genetic variability of the progeny is sometimes undesired, for instance, like in crop seed production. One reproductive strategy that is very interesting to have less or no variability in the progeny is indeed apomixis. So apomixis is a plant reproductive strategies that allow the formation of seed without fertilization. So the seed that are produced, they are clonal, identical between each other and also identical to the mother plant. And this could happen through different mechanisms. Here is just a summary of the basis of apomixis, where I compare the apomictic reproduction to sexual reproduction. As you can see, the first step is to obtain and reduce female gamete formation. And this could be achieved making gametes without meiosis. Meiosis, I remind you, is also a process that introduces variability through recombination. Those reduced female gametes then can go from one side to develop an embryo without fertilization through a process named parthenogenesis. And also the central cells can also give origin to the endosperm without fertilization. So in other words, we can obtain seeds without fertilization and without meiosis. And for this reason, all the seeds obtained with these strategies are between them identical. This is extremely important reproductive strategies because not only for seed production, but also because it's a method to have one-step fixation of any trait. Is a method to get faster development of new variety, and also that also is important to reduce the losses of a poor pollination. For this reason, for all these reasons, there are many groups in the last decades that have invested a lot of time, energy, and money to try to understand the genetic basis of apomixis. That is very quite a rare reproductive strategy in nature. Indeed, only 0.1% of the flowering plant present this type of strategy. And also, I have to say that apomixis is absent in the major crop. In Milano, we are also involved in understanding the basis of apomixis in collaboration with KeyGene. We are working on the understanding of the first step of apomixis, so the step in which the female gametes are formed without meiosis. But about that, we will have a nice talk after I end with mine. So to conclude, I just list some interesting let's say, topics on plant reproduction that we are carrying on in our department at the University of Milano. But I want to stress that there are really several open questions in plant reproduction, sexual and asexual reproduction, that have still to be addressed. And there are many problems to be solved. So a lot of opportunity in this field. And the last slide, because you are all here, means that you are all interested in plant reproduction. I would like to make a short advertisement for the conference that will be taking place in Prague in June on sexual plant reproduction, that will also include a workshop on Apomixis. So thanks for listening, and ready for questions.
Well, thank you very much, Lucia, for your introducing presentation. Nice presentation, clear presentation. We don't have questions from the audience yet. We have more than 400 participants. I would like to invite all the participants to write down your questions. But in the meantime, I have two challenging questions, I think, for you. How do you think that the pollen grain will find the egg cell, and how will it find the opening of the ovule? Is there anything known about that? Can you say something about that?
Yes, sure. There is a lot of research that has been done in the past and are still under investigation that had allowed to identify pollen and female factors that are extremely important and required for interaction between the pollen tube and the female tissue that allowed a successful fertilization. So each single step from the interaction between the pollen and the pistil and all the steps that lead to the formation of the pollen tube and the deliverable of the two sperm cells inside the ovules are strictly under genetic control, and many of the factors that are involved in this process have been identified. And so there are already, for sure, many questions still to be answered, but many different factors and network has been already described and characterized.
Yeah. And because of the fact that the egg cell is carefully hidden and protected, also in the ovule and in the carpels, deep inside the pistil, the whole process of fertilization and development of the egg cell into a zygote and the zygote into a seed is difficult to follow. What type of tool would you like to have? What type of tool do you dream of, which will give you a breakthrough in studying and let's say understanding this process of sexual plant reproduction?
So, of course, as I said, this process is extremely complex because, of course, the plant receiving the pollen has to check step by step that the gametes that are carried inside the female tissue, they have the proper characteristic. And to be able to follow all these steps is nice to develop. And indeed, there are some confocal that already some confocal microscope that are already available that allow to follow this process in vivo. So you can really follow each single step, and from the interaction with the stigma, that is the first female tissue involved in this pollen, so male and female interaction, till the fertilization. So let's say that for me, it would be the best is to follow in vivo. So to follow through, for instance, spatial microscopy tools to be able to see step by step what is going on during this important process.
I see two questions from the audience. Maybe the last question, which is, when Apomixis will work on crop, will it be the end of a seed company? No more hybrid seeds. I think we will keep that for the panel discussion. But there is also, maybe you know something about that, a question from Abhilash. Is there any natural mutant that exists in coffee that follow Apomixis? And otherwise, we also keep that question for the panel discussion.
Okay.
Maybe somebody can take a look at that and know something about that. Yeah. Okay. Thank you very much, Lucia, for this very nice presentation. And we continue with the next speaker, which is Rik Op. And Rik Op started at KeyGene as a researcher in 2012. And his current role is team leader and project leader at KeyGene. He received his PhD in plant microbe interactions from the Wageningen University. And Rik has worked on a range of topics, mainly in the field of plant reproductive biology. This resulted in multiple patent applications and also into scientific publications. He likes interdisciplinary approaches and to develop cutting-edge technologies for application in plant breeding. In his team, breeding support technology, Apomixis is one of the long-standing topics, and KeyGene is already working on Apomixis for more than 15 years. Of course, he's very proud of his team to have reached the milestone of cloning the PAR gene, and the title of his presentation is Booster for Apomixis, PAR and DIP genes. So, Rik, the floor is yours.
Thank you, Arjen, for this nice introduction. So, hello, everyone. I would like to say a bit more on our work on Apomixis. And as Arjen already indicated at KeyGene, we have been working on the topic for a very long time. And our overall strategy is based on the reverse engineering of natural Apomixis. As Lucia mentioned, it is not very common in nature, apomictic reproduction. But you can find quite a few species when you look around, at least here outside, for example, dandelion, hawkweed, or brambles. We would like to understand the natural mechanism of Apomixis and clone the underlying genes, with, of course, then the final goal to transfer this to crops. And our favorite model is the common dandelion. This model is a very good model to study Apomixis because sexual and apomictic seeds have a similar development. For some apomicts, this is not the case. Also, it has a relatively small genome size and low ploidy levels. And the material we collected quite nearby. It is material gathered in the Netherlands, and in the fields you can find sexual and apomictic plants next to each other. Having then sexual and apomictic material of the common dandelion, then another advantage of the common dandelion comes into play, because the apomictic mechanism in dandelions is really restricted to the female meiosis, which means that the pollen formation, so the male meiosis, is normal. So you have normal reduction, and that actually allows you to use an apomictic plant as a father in a cross. This eventually will also facilitate breeding applications using this type of Apomixis. So when you make these crosses, you can look in the offspring whether the developmental elements of Apomixis segregate. The two elements I would like to discuss today are the plospory. So this is the first part, as Lucia indicated. The first step is then when you have no longer a normal meiosis, but a process that leads to the formation of female gametes that have the same genome copy number as the mother, so are not reduced, and also have undergone no recombination as you would normally have in meiosis. This process is the first step, and then the second step is the development of the egg cell into an embryo without fertilization, called parthenogenesis. And having made these crosses, it became clear that they were represented by two unlinked loci called the dip and the PAR locus. And in case of the apomictic dandelion, there are 3 alleles, so 1 apomictic and 2 sexual alleles in these loci. The next step was making use of the power of mutagenesis. So mutant screens were used to screen for the loss of the individual elements of Apomixis, so the loss of diplospory and the loss of parthenogenesis. And in dandelion, these phenotypes, as you can see by the pictures at the bottom, can clearly be distinguished from the wild type. So a loss of parthenogenesis, they make no embryos, so the seeds are empty. You can see that they become whitish. Whereas in the loss of diplospory, parthenogenesis is still present, so few seeds will be made because diplospory actually then reverts the process towards normal meiosis. So having screened these mutant populations, we were able to find a minimum deletion window, and thereby, of course, using genetic markers, identify the two loci for diplospory and parthenogenesis in dandelion. They are about 500 kb in size, and they have some 50 genes each. We used long-read sequencing technology to sequence few of these mutants, and the illustration shows in yellow the deletion found in one of these mutants. And as the deletion areas have different sizes from the various mutants we picked up, you can narrow down the window where you should look for the causal gene. To find the causal gene, we moved two different strategies as they were depending on the technologies available at the time we worked with it. So diplospory have been working on for quite some time, and we followed with another screen of mutagenesis, but then chemical mutagenesis and resequencing of the genes in the locus to look for polymorphism specific for the apomictic allele. And in case of cloning the parthenogenesis gene, we followed a gene editing approach targeting the individual genes in this locus. And one of the questions we had was whether there are unique apomictic genes or that they rather are modified sexual genes having otherwise a different function. So the gene for diplospory we managed to clone is called VPS13. It's a highly conserved gene, and it has also been described in Arabidopsis as the shrubby mutant. It's a huge gene, so it's about 400 amino acids and having 46 exons. And this is currently still under investigation. Amongst others, we work on this with the University of Milan and the group of Lucia, Colombo. For now, I would like to focus on the cloning of the PAR gene. So for the cloning of the PAR gene, we used gene editing to target several genes in the locus. And as there were about 50 genes, we were not able to target all of them at once, so we made a selection based on conserved polymorphisms in apomictic alleles and also a bit of annotation gazing. And eventually everybody needs sometimes a bit of luck. And we were quite lucky actually that among our first selection, we were able to find a gene that and that is actually the only gene we found in that locus, that when you knock it out, by causing a frameshift deletion with gene editing, you see the phenotype as I had in the earlier slide, so this whitish seeds that are sterile because no embryos are formed. You can see on the Petri dish that the seeds did not germinate compared to the control. But as this process these plants have undergone regeneration in tissue culture, you may think it is due to a sterility or female sterility effect. So in order to rule that out, we make a cross as if it would still have diplospory intact, it would make unreduced non-recombined egg cells, and you can do a normal fertilization. You would again expect to get tetraploid plants out of that. And these crosses indeed yielded tetraploid plants. So having the 3 copies of the apomictic plant with 1 reduced pollen copy of the sexual plant giving rise to tetraploid plants, which is confirmed by the flow cytometry result here, and it resulted in fertile offspring. So having knocked out the gene and getting this phenotype, this was a very nice piece of evidence that this is the gene for parthenogenesis in the common dandelion. So what does the gene look like? The PAR gene is a zinc finger with a single specific zinc finger domain, which is an indication that it may be DNA binding, and it has an ear motif, which is an indication that it may have a repressive function. Of course, we looked for specifics of this gene. And actually, our partners at Wageningen University also performed extensive transcriptional analysis by RNA-seq on Apomixis in dandelion and comparing this to sexual species. And one of the things we found there, is that it is a gene only expressed in the egg cell, in the Apomix, but not in a sexual species. And another good clue then on how this may work came from looking at the promoter regions. As only in the apomictic allele, we found a transposon insertion just upstream of the start codon, which may indicate that this alters somehow the regulation of this gene. Then, having the egg cell expression and an alteration in the promoter, we had an experiment where we tried to express the PAR gene in the egg cell in dandelion in the mutants that we made with the gene editing. So that mutant has no parthenogenesis. We could restore the phenotype of parthenogenesis by expressing the dandelion PAR gene driven by the egg cell specific promoter von Arabidopsis, meaning that expression in the egg cell can give rise to parthenogenesis in common dandelion. Then, as our final goal is introducing it to crops, we move to a close relative of the common dandelion, namely lettuce. It is a member of the same plant family of the Compositae, and the same genetic construct where we express the PAR gene of the common dandelion using the egg cell specific promoter in lettuce, we saw the following result. And here we were very happy to see that in controls, we see actually nothing, which is good because we removed all the pollen, so no embryo can form. But when the construct was present with the parthenogenesis gene, we could see the start of the development of embryos. So the parthenogenesis gene can induce embryos in a sexual diploid crop. This is a very nice proof of concept. However, of course, further fine-tuning will be needed, and we are looking into that at the moment. So how then would this all work? We have come to the following model for the functioning of the parthenogenesis gene. In a normal sexual plant, embryogenesis is suppressed in the egg cell because it is waiting to be fertilized. And there is a repressor present for embryogenesis. The sperm cell then, when carries transcripts or proteins of the parthenogenesis gene to the egg cell, and when they fuse to form the zygote, this is then triggering the formation of the embryogenesis program in the embryo. Whereas in apomictic reproduction, we know that the parthenogenesis gene is expressed in the egg cell, and thereby it can already actively inhibit the repressor of embryogenesis, and by inhibiting the repressor, you get a spontaneous start of the embryogenesis program. So, the parthenogenesis gene from the common dandelion can induce parthenogenesis in a sexual crop. There are PAR gene homologs present in most of crop species, and the sexual or normal function of the parthenogenesis gene is to initiate embryogenesis after fertilization. I would like to thank the KeyGene team for all their hard work and actually very long-standing work on this topic of Apomixis, and also our partners from Milan, from KAAS, from Wageningen University, and from the Lincoln University for making this all into the success of cloning these genes. And thank you for your attention.
Well, thank you very much, Rik, for your very clear presentation. Nice results. A couple of questions from the audience. Question number one is, do transposons jump and move around in the genome during apomictic reproduction? Do you know that?
If they move around during apomictic reproduction, well, I think you do not get recombination. If you get activation of transposons, I personally would not know for sure. But what we have seen is that Apomixis, the genes and the loci in the region where the Apomixis genes are, we see conversions. So events that indicate that parts of the sequence have exchanged between the alleles. The underlying mechanism we do not know, but these are observations we made.
Okay. Another question from the audience, from Jose. Are the PAR and DIP locus are they on the same chromosome?
No, they are not, and we found that they have in this early genetic studies that they are independent loci and they segregate independently. Yeah.
Another question from the audience: did you already try to use the promoter PAR construct in monocots?
We haven't embarked on that. And these experiments, we've chosen to stay first closer to the dandelion, but of course we did find homologous sequences in monocots. Yeah.
Okay, interesting. The last question is from Stefan. Apomixis is an end station unless there is a breeding mechanism behind the tool. He states there are examples in tropical grasses that currently are used. How does the technology of KeyGene relate to this approach? Do you foresee the same type of application?
Yeah, so as I mentioned in the beginning of my talk, the apomictic system of the common dandelion is quite unique that it is restricted to the female gametes, which means that an apomictic plant still produces normal pollen. And this pollen you can use to fertilize other plants that are not apomictic, for example, to make new variation. And you can cycle this process. Once you find among offspring that still carry both Apomixis genes, a plant that you are interested in, you can keep on clonally propagate that one via seeds and make it into a product. So it is, to my view, not an end station. You can continue to make crosses and to add variation to your apomictic pool.
So breeding via the male line and apomixis via the female part.
Indeed.
Right. Okay. Well, thanks a lot. We'll move on to the third speaker of today, Tim Sharbel. Focus of Professor Tim Sharbel's, welcome Tim. Focus of Professor Sharbel's research group at the University of Saskatchewan in Canada is the evolution of naturally occurring asexual seed production in plants. This research encompasses population genetics, evolution, high-throughput phenotyping, various genomics technologies, and also functional genetics. Tim has attracted additional interest and also funding from both industrial parties and private sources for his research. He has 15 years of research background in Germany in apomixis and reproduction in the medical plants, St. John's wort, Hypericum perforatum for dementia treatment research, and German chamomile, I don't know that one, but for digestive applications. His interest in medicinal plants has remained, and he also started working on genomics in medicinal cannabinoids nowadays. And the title of his talk of today is Induction of Apomixis in Brassica using tools development from the apomictic Bucera complex. Tim, the floor is yours. Welcome.
Thank you. So I'd like to thank the organizers for inviting me to give this talk. It's wonderful. I'm very, very, very excited to be here. I'd like to congratulate KeyGene and Eric and Peter and everyone else. Amazing to see this coming out. As I was in grad school, my first readings of Apomixis were, based upon a lot of Peter's work in Taraxicum. So it's beautiful to see all of this coming full circle. So we have, Lucia already brought up sort of the points of why apomixis is so important. So I'll just brief over this really quickly, right? So apomixis it's a very interesting topic from evolutionary biology, trying to understand why organisms switch from sex to asexual reproduction. Why is it important for breeders, right? So it really is all about speeding up breeding and producing locally adapted varieties and the introgression of complex genetic traits and fixing those traits, right, in crops. And then for farmers, right, there's the reduction of time and costs for seed production and really the fixation of hybrid vigor. So this is a true future breeding technology that is needed everywhere. And so I've spent my life working on Bucera now for over 20 years. And so there's more than 100 species in this group. This is just a nice picture which shows you some of the diversity that we see in the plants themselves and the flowers and the reproductive tissues. And so you can see there's a great deal of variability, which we can actually mine and understand from natural populations. I started working on this at the Max Planck Institute, and I would extend a very significant thank you to Tom Mitchell-Olds, who was my supervisor at the time, more than 20 years ago. And he's the one that introduced me to Bucera. And so it's closely related to Arabidopsis. So it's a wild brassica plant. And obviously it's close to brassica crops. It's got a very reasonable genome to work with, right? So between 170 to 230 megabase pairs. And it's found throughout North America and Greenland. Very quickly, Bucera reproduces through sexual means, and we all know how sex occurs, right? So we have fertilization to make an embryo and endosperm with a diploid to triploid ratio, and this is very normal in sexual plants, obviously. But in Apomix, so we have Diplosporus Apomixis in Bucera, and so we have the production of a meiotically unreduced egg cell in females. We do have pseudogamy, right? So the male function in Bucera produces both haploid and diploid pollen. And what we find is that most of the time we have fertilization of the tetraploid central cell to form balanced apomix with an embryo ratio of a diploid embryo to a hexaploid endosperm. But very importantly, and I want to point this out because it is key to some of our research, is that we have a very low frequency of unbalanced apomix, right? So we have fertilization with haploid pollen to produce embryos, diploid apomictic embryos, but pentaploid endosperm, which are unbalanced, obviously. We have found marker genes in the past, so in my lab, and so two ones that we have published are Apollo. So this is associated with apomyosis, so in the female function, the production of meiotically unreduced egg cells. Very quickly, there is an apomictic allele, which is found exclusively in apomictic individuals. And that allele is expressed in ovules when there's no expression of that gene in sexual ovules. So the apomictic allele is expressed in the apomictic ovules. And furthermore, we've identified a second marker gene, which is associated with meiotically unreduced pollen formation, and this is referred to as upgrade. It's a very complex gene, which is essentially a chimera between different kinds of genes in the genome. But very importantly, these two markers have very, very high association with Apomixis. So Apollo has a 98%, upgrade has a 96%. When you look in natural populations, we find both factors in about almost 80% of the individuals we look at. And very importantly, as I said, these are proxy markers for us, right? Very interestingly, this is some unpublished work, which will hopefully go to journal very soon. But what we've been working with Martin Lysak and Terry Mandakova in Brno, some very beautiful work. And what's very interesting and relatively unexpected is that paracentric heterochromatin seems to be involved with the evolution of the Apomixis genes we've discovered. So for example, Apollo is found on chromosome 2, and you can see sort of a nice, we've got a chromosome 2 fish marker here, right? And we've got Apollo here, and you can see that they're together. And interestingly enough, not on the het chromosome, which is a divergent chromosome we see in apomictic Bucera. Upgrade, very interestingly, is found both in sexual plants, so one allele of it, but very interestingly in both diploid apomictic Bucera and diploid apomictic Bucera with an extra chromosome. This is very interesting, and you'll see that upgrade actually maps to this apomictic chromosome, to the het chromosome. And this follows a lot of observations made in both plant and animal asexual plants and animals, because it seems to be a very good place for apomictic or asexual factors to segregate themselves into. They're safe places in the genome. Upgrade 2, of course, we have been going more and more deeply into the function, right? So upgrade 2, we've figured out is as a potential torpedo regulator of microspore development at meiosis. And I'm not going to show you any Apollo data, but it really does seem to be so when we look at Apomixis in Bucera, there seems to be kind of a continuum from pollen all the way to the ovule. And so we're very interested in the signaling and kind of the chain of pathways that go really, and it seems to start in the pollen population on the way to the apomictic ovule. So very quickly, we're working on a 5-year plan to get Apomixis into brassica. So, and I'm in Saskatchewan, obviously, and canola is a huge crop here. So we've been funded by the canola researchers and farmers, and we're also been funded by the government. And I'm just going to show you 2 parts of this pathway to get to canola. We've got crosses going on one hand, and they're both interspecific and intergeneric crosses. We've got back crosses going on between sexual and apomictic buccara. And then in the second pathway, we're taking a completely genomics approach, sequencing approach to identifying factors, and then putting the idea is to put them into a cassette to transform into brassica eventually. And taking these 2 things together over the next 5 years, hopefully will get us into, to developing apomictic brassica. So in terms of the crossing, this is the work of Dr. Martin Mao in my lab. So he's been with me now for almost 15 years. And Martin started a crossing project about 6 years ago when we were in Germany, and then we subsequently moved it here. To make a long story short, haploid pollen seemed to be the very important factor, which account for the spread of Apomixis in natural populations. And so what we have, and I'll show this to you more in detail in the next slide, is essentially when we look at different species of buccara, we've got diploid sexual outcrossing populations, and then what we have is a very complicated gene flow, which goes all the way to diploid balanced apomix, triploids, and so on, right? So there's this massive gene flow going back both towards apomixis, but also coming back towards sexuality. And people kind of underestimate these effects on natural apomictic populations. After doing many crosses, we published this paper last year and showed that there really is a lot of variability that comes from crossing apomictic individuals onto sexual individuals. And what we've been doing is we've been crossing balanced apomix, which produce diploid pollen, and we've been crossing unbalanced apomix, which have haploid pollen. We've been crossing these onto multiple species of sexuals. And the take-home message is essentially you can generate triploid apomix, which come out of this, which we see in natural populations. They're for the most part sterile, right? But importantly, that the unbalanced apomix and the haploid pollen can produce a new apomix when you cross them onto a sexual background. They can produce a brand new diploid apomix in 1 generation with all of the components. And if you further cross those things, you'll see that we still have an array of different kinds of offspring that are formed. But very importantly, we have a large percentage of meiotically unbalanced apomictic diploids that are coming out of this. So based upon these data, Martin has been working very hard on creating synthetic hybrids. And so we've been, so once again, the crosses are between balanced and unbalanced apomix and sexuals. And to make a long story short, so this was published once again last year. We've been working on natural selfing. So very importantly, understanding, what the baseline is for kind of normal sexual fertilization, right? Like what's the normal state? It's not 100%, right? You actually have to kind of go into sexual plants and measure that. And what we see, in fact, is that we have great differences between different sexual plants in terms of their ability to form sexual seeds. But more importantly, we've got intraspecific outcrosses and we've got interspecific crosses going on. A great deal of these things running in our greenhouses here. And clearly we have a lot of material that we're presently screening. So looking at the screen, so we've got where we are is we're aiming for 2020 or 22 to be into the final back crosses, and we can do things like segregate. We can segregate parthenogenesis. We can segregate apomeiosis and so on. And the second approach we're taking is, so this is within buccara, but then we're also taking an approach where we're crossing buccara onto a bridge species. So it's this is wasabi actually, so diplotaxis, and we're trying to get crosses apomixis into diplotaxis, followed by the transfer into brassica. The final part then is just the Apomixis cassette, and this is done with Philip Rigaud at a company called Geidel, which is in Quebec City. And so what have we done over the last few years? We have a completely finished chromosome, telomere to telomere, finished quality reference genome using different tools from Guidel. So we've used multiple levels of DNA sequencing, including optical mapping. And very importantly, if you look at the LTR assembly index, so this is an index that was published in one of the Nature journals a couple of years ago. And if we look at the assembly index and we look in some of the important crops here like rice and Arabidopsis, which have been very well sequenced, you can see that the index, our assembly index, is even much higher than those. So we've got a super good genome to which we can compare our data. And then the final slide is that Philip essentially has taken the approach, he's taken the bioinformatics approach and doing the work of a geneticist. And so we've put in 3.5 billion fragments of our own sequencing. We've combined this with public sequences. It amasses to about 30 individuals of sexual and apomictic buccara, 7 different species. We've added degradome information to this. We've added a phylogenetic context to this, and so on and so on. So these comparisons between sexual and apomictic individuals, very, very thoroughly done, have independently identified apo-specific duplications. Apollo, we identified independently. We found apo-specific genes and regions. And once again, we found upgrade independently, right? So Philip has bioinformatically done the work of a geneticist. And very importantly, we have a lot of apo-specific variants. So, and these are all now in our, in the laboratory. And what we're doing now is essentially putting all of that information together. As I said, we can segregate the different components of Apomixis, and so as we proceed this year, we will be beginning the mutant analyses of the different genes that we have. So with that, I'll stop. This has been fun. This work has gone on now for more than 20 years. It's been funded by many, many different organizations, both in Europe and in Canada. And so I would like to thank them. I would also like to thank everyone. And this is only a small set of people who have really worked on this over the last 20 years. And so really kudos to everybody for all the help. We're getting there. And once again, really happy to see KeyGene advancing with this as well. It really gives us heart to know that we're on the same pathway. And so I'll stop there and take any questions if anyone has any.
Thanks, Tim, for your wonderful presentation. I have questions from the audience.
Sorry, I'm just stopping to share my screen here. There we go.
There's a question from Andre for Tim. Have you made or obtained any successful interspecific crosses with canola already?
We have crosses, yes, and we've actually we can demonstrate, we can confirm the crosses with markers. So we have buccara-specific markers. And right now, we are also examining chromosomal integration with Martin Lysak and Terry Mandakova and Brno. And we're growing the seeds and trying to figure out if they're producing anything that looks like Apomixis.
Okay, and he continues with another question. Could such interspecific hybrids induce haploidy in oilseed rape? Oh,
maybe. I don't know. I mean, we don't see any haploid parthenogenesis in buccara, right? So, let's just say it's not in the spectrum of phenotypes we see in buccara. So, you know, I guess it's unknown whether or not that could essentially happen in canola as well. I don't know.
And there's also a question from Joanne. Any thoughts on criteria for the choice of a bridge species?
Yeah, the bridge species. So this is Martin Mao's work, but basically looking for things that have kind of similar chromosome numbers and similar genome size is kind of the approach. And that's why we chose this particular species. And once again, we do have crosses going, and we have been able to assess that some of the crosses are working using markers. But we don't know the extent of it right now. And remember, you know, we've got from the genomics, we have all of the Apomixis markers, right? And unfortunately, I can't tell you more about them, but it's a workable handful number of genes, right? And so really the question is, can we get all of those into our crosses as well? Or is there some segregation problem with those particular markers because they're found in pericentral heterochromatic regions and so on?
Yeah, that's also a question from my side. My impression is that you think that Apomixis in buccara is evoked by multiple genes.
Yeah, and I can tell you, yeah.
At least two, Apollo and
yeah. And I would say it's comparable to what has been discovered in other species like Taraxacum and Pennisetum when a lot of sequencing has been done in these hemizygous regions. Let's just say it's the same kind of handful of genes in terms of numbers.
So you think 5 to 10 or so, or?
I would say more like a kind of 20 to 40, depending on. But once again, we're screening them, right? And it's very interesting because when you look at the genes and that are part, that are Apomixis specific, what we realize is it's not only genes associated with expression of the standard traits that we think about in Apomixis, right? So we think about apomyosis, we think about parthenogenesis, and so on, right? But when you think about it, there's got to be you need other factors. And I see this kind of like a Y chromosome analogy, right? You need other factors, for example, to turn gene expression into the female tissues from male tissues. You need, well, I won't go on with these because I know you have time, but essentially you need more than the factors to have a stable apomict than just factors associated with the different components that we typically speak of. That's all I wanted to say.
Interesting. Well, once again, thank you very much for your wonderful presentation. We continue with the presentation of Kejian Wang. He is a professor from China National Rice Research Institute of the Chinese Academy of Agricultural Sciences. During his PhD in the Institute of the Genetics and Developmental Biology of the Chinese Academy of Sciences, he mainly focused on investigating the fundamental mechanism of meiosis. After setting up his lab in 2013, Professor Wang became interested in developing novel genome editing systems for crop improvement and establishing synthetic apomixis in hybrid crops. In 2018, Kejian's lab successfully established the synthetic apomixis strategy by genome editing and obtained the clonal seeds in hybrid rice for the first time. And the title of the presentation of Professor Kejian Wang is Synthetic Apomixis in Hybrid Rice. So Professor Wang, thank you for speaking, and the floor is yours.
Okay, thank you very much for your nice introduction. And before I start, I would like to thank KeyGene for organizing the webinar about apomixis. Today, I will talk about synthetic apomixis in hybrid rice. As we all know, catalysis is a common phenomenon that has been widely used in agriculture. For example, in this picture, the hybrid rice variety and CY84 in the middle and displays much increased vigor compared to its homozygous parents. The hybrid variety displayed an elite traits in fields. However, the elite traits were segregated from F2 generation, and therefore, hydrolysis application requires yearly and hybrid seed production, which is extremely complex and significantly increase the seed costs. And to simplify and maximize the application of hydrolysis, it has long been proposed to fix hydrolysis by introduction of apomixis in rice. If apomixis occurs, the hybrid plants will produce clonal seeds, so the hydrolysis will be preserved in each generation. Despite several decades exploration, the apomixis rice has not been found in nature. The left is a picture showing normal sexual reproduction. The red and the blue lines indicate the homologous chromosomes from two parents. During meiosis, the homologous chromosome will undergo recombination and produce recombined haploid gametes. During fertilization, two haploid sperm two haploid gametes will fuse, and the diploid recombinant embryo will be generated. So To achieve apomixis, both meiosis and fertilization must be passed together. Based on this, we propose a strategy of synthetic apomixis in hydrolysis. The step one, bypass normal meiosis and produce clonal diploid gametes. And step two, bypass normal fertilization and resulting in the parthenogenesis of the egg cell. By combining the two steps together, theoretically, the hybrid will produce clonal embryo. We first test the feasibility of the first step, production of clonal diploid gametes in hydrolysis. Actually, in 2009, Raphael's lab found that if 3 genes evolved in meiosis were OSD1, SPOR11, and the record were co-mutated, and in adults, the triple mutant Miami will generate clonal gametes, and the ploidy will be doubled in each generation. To test whether Miami also works in hydrolysis, we selected the 3 genes, record pair 1 and OSD1, for simultaneously generated in CY84, the hybrid variety. After DNA sequencing, the triple mutant were undefined and used for our analysis. We next investigated the meiosis of both wild type and the Miami mutant. In the wild type, 12 pairs of homologous chromosomes will undergo pairing and recombination. After 2 rounds of cell division, tetrads will be produced. On contrast, in Miami, the homologous pairing and recombination were totally disrupted. And after only 1 round of cell division, only dyads will be produced. Next, we conducted fixed experiments to determine the ploidy of the Miami, also the wild type. The blue colors indicate the dependent DNA, and red indicated the 5S RNA on 11 and chromosome. As we can see from in the wild type, only 1 signal can be found, but in Miami mutant, 2 signals were constantly observed, indicating the gametes were diploid. We next investigate the fertility of the single mutants and the triple mutant. Both the record and the pair 1 mutants were completely sterile, and no seeds were found. But in the seeding set of the Miami triple mutant, we can compare. It shows almost the same phenotype as the wild type. These results differ from previous study in homozygous varieties, suggesting the background may affect the seeding set. The offspring of the wild type and the Miami were also investigated. The progeny of Miami should increase the grain size and elongated own length. All those phenotypes are typical characteristics of tetraploid rice. Next, using 10 indel marks located on chromosome 1 and chromosome 8, we identified the genotype of the progeny of Miami, and found that all the sides of those offspring were heterozygous, indicating that the genotype was fixed in Miami. This work improves the possibility of production of clonal gametes in hydrolysis. It works pretty well, and then we want to test the possibility of parthenogenesis of the egg cell in the hydrolysis. Before our work, there are 3 strategies to induce parthenogenesis, and also induce haploids. The centromere-mediated genetic and ectopic expression of baby boom-like genes. Also, the mutation of MTR genes in males also can induce haploid induction. And because we want to this gene, the second stretching needs a ectopic expression of BBM genes, and this method only need a mutation of one gene. So we test this possibility in rice. And the homologous genes of MTR also exist in rice. We therefore edited the candidate gene to test the feasibility of haploid induction in haploid rice. We found that the MTR mutant displayed normal vegetative growth, but the seeding cell rate significantly reduced from about 80% to 11%. We harvested all the MTR seeds and 12 markers on each chromosome were used to determine the genotype of the stream. During normal sexual reproduction, because of the occurrence of recombination and fertilization, the theoretical frequency of homozygous recombinant implanted bleeds will be pretty low, about 0.024%. And actually, we didn't find any plants that were homozygous at all. On contrast of the 214 progenies of MTR, 11 plants were found to be homozygous at all 12 marks. Those plants were potential haploid or double diploids. The ploidy of plants were investigated using flow cytometry. Of them, 2 were undefined to be double haploids, displaying normal growth, and the 9 plants were undefined to be haploids, showing dwarf and stellar phenotype. Next, the whole genome of those plants and the parent plants was resequenced. The red and blue colors indicate 2 parents respectively. Yellow color shows heterozygous F1 hybrid. By informatic analysis, revealed that yellow colors were observed in many chromosomes of I read. On contrast, the haploid and the double haploid plants were homozygous at all chromosomes. That is, no yellow colors were no yellow colors. Together, these results imply that those plants are possibly generated from the personages of the egg cells. After testing the feasibility of 2 steps, we next try to combine 2 steps together, and want to know whether synthetic Apomixis can be achieved and whether we can get clonal seeds. We added all those 4 genes mentioned about, and investigated the meiosis. And this is the mutant was named fixation of hybrids, fixed for short. The fix displayed the same meiosis phenotype with the Miami, that only 1 round, no recombination, and only 1 round of cell division and produce dyads. We also observed the phenotype of the mutant. This is the Y-type hybrid, CY84, and this is the fixed mutant. It displays normal vegetative growth, but the seeding setting rate is also reduced significantly, only about 4.5%, which is even lower than that MTR single mutant. Next, using flow cytometry, we found of the 149 seeds, 7 plants were found to be diploid, and the rest are tetraploids. We also conduct a resequencing of all those diploid and triploid, and we found that all those plants displayed heterozygous genotype at all those sites. These results indicated that those plants and the ploidy and the genotype are fixed, indicating that they are clonal seeds. Next, many people are very interested in whether the phenotype can be inherited stably to the next generation. So we planted the plant and the fixed plants in different generations, the first generation and the second generation in glassroom, and the third generation and the fourth generation and the fifth and the sixth generation. We found that the fixed mutant displayed a normal vegetative, but and other phenotype remains stable, and it also shows a stable low seed seeding rate. We also retained the seeds in each generation and planted them in the same paired fields, and found that they displayed nearly the same phenotype, indicating that the apomixis phenotype can be inherited stably from generation to generation. Now, our ongoing work, now we are looking for the germ plants and the genes that increase the seeding cell rate and haploid frequency, because we found the seeding cell rate and the haploid frequency was different in different backgrounds. So they must be controlled by some genes. I want to isolate those genes and reveal the mechanism of the mechanism. Also, we want to clone more genes involved in past synergies of the egg cell. Also, we also testing the stretching in other crops. Finally, I would like to thank my collaborators, Rafael Mosher, Dukan, Mingo, and Jianlong, and also my lab members, also the funding support. Finally, thanks for your attention.
Thank you very much, Kejian, for your very nice work. Due to time, we have only time for a few questions. The first question was about the germination of your apomixis plants. Was that normal or was that reduced?
The germination is the same as the white type.
Okay. And a quick question. 4.4% haploid induction is low. Is that because of the knockout of the MTL gene also induces aneuploids?
Yes, it's mostly because in our background, the haploid variety, the Miami displays nearly normal seeding cell rate as the white type, but the MTL was pretty low, so we think it's mainly affected by the MTL. And now, so we want to isolate more genes and like MTL or modify the MTL gene to increase the seeding cell rate.
Okay, and the last question is from Robert. What about the endosperm formation in your apomixis rice? Does that occur autonomously, or how does that go?
The endosperm is normal in those seeds, but there are many clones are empty. That means their endosperm is disrupted. So I think the low seeding cell rate is caused because of the MTL and the endosperm. Or most clones cannot form normally those MTL mutants.
Okay.
But only some can form normally.
Okay, thank you very much for your presentation.
Thank you.
We have to continue, and we'll continue with the panel discussion now. and I would like to start with introducing the panel. First of all, Charlie Underwood. I would first like to congratulate him and his wife for the birth of his son. I think it was yesterday. Charlie, well done. And I hope everything is okay with you and your wife and your newborn. Charlie studied at the University of Oxford, received his PhD in plant genetics, epigenetics and genomics under the supervision of Rob Martinussen. Charlie is a research group leader now at the Max Planck Institute for Plant Breeding Research in Cologne. His group is interested in the factors that control sexual and asexual reproduction in plant species. He and his team explore how different elements of plant reproduction, including meiosis and fertilization, have been molded and adapted over evolutionary time. He has been working at KeyGene also for a couple of years, and there I got to know him as a very dedicated, very smart scientist. So I expect a lot of results from his work at the Max Planck Institute. The second member of the panel is Rik Schranz. Rik is a professor of biosystematics at Wageningen University and Research since 10 years already, since 2012. He was an undergraduate at Cornell and received his PhD from the University of Wisconsin, Madison. He was then a postdoc with Tom Mitchell-Olds, first at the Max Planck, then in Duke University. During his postdoc, he began investigating various aspects of Apomixis in Bucera, often in collaboration with Tim Sharbel, Professor Tim Sharbel. When Rik became assistant professor at the University of Amsterdam in 2007, he started working also on other plant species, Brassica, Solanaceae, Asteraceae, working together with Kitty Vijverberg on an NWO program. He was involved in the PAR tool project, cloning and characterizing the PAR gene in Taraxacum. Looking to the future, Rik is excited to investigate how the identified parthenogenesis gene functions and how it can be, how more broadly the gene family is and the associated pathways have evolved across the Asteraceae species and beyond to more distant crops. The final two members of our panel are Professor Charlie Spillane. He's the chair of plant sciences and also the director of the interdisciplinary Ryan Institute for Sustainability at the National University of Ireland, Galway in Ireland. Charlie's Genetics and Biotechnology Research Group works on plant and agricultural biosciences for sustainable development. He obtained his PhD in plant molecular genetics at Smurfit Institute of Genetics in Dublin. And he also worked on plant genetics and epigenetics at the John Innes Center, Cold Spring Harbor, and in Switzerland at the Friedrich Miescher and the University of Zurich. Charles also worked extensively for the FAO and the CGIAR, providing policy and technical advice on agricultural biotechnology. And now he is founder and director of the award-winning master's degree on climate change, agriculture and food security. And he is also the chair of Irish Forum for International Agricultural Development. Then the fourth member is Peter van Dijk. He is here at KeyGene in Wageningen. He is a distinguished scientist at KeyGene. His main research focus has been on plant reproductive systems. And for the last 25 years, Apomixis in particular. He received his PhD in population genetics from the University of Groningen. From 1991 onwards, he was a scientist at the Netherlands Institute of Ecology. And in 2006, he joined KeyGene. And there he continued his work on cloning and identifying Apomixis genes in dandelion, resulted in the identification of the PAR gene in 2022. And he was involved in 29 scientific publications. I should also mention that one of his biggest hobbies is to investigate and study and publish on the life and works of Gregor Mendel, who, by the way, also did his R&D in not only genetics of pea, but also in some apomictic plants. So I think we have a very nice panel. It took me quite some time to introduce them because they are very experienced, but that's very nice. And I would like to start with a general question. What do you think of the presentations? Did you see anything new? What did impress you? What did it make you feel? Who wants to say something about that? So Charlie, Eric, or Peter, can I invite you to react on the presentations? Don't be shy. Eric, what is your? Charlie. Yeah.
Yeah. No. I think it's wonderful. Firstly, thanks for the invitation to be here today. I think this seminar is really timely because we're getting to see the culmination of decades of work by very talented and committed researchers in the Apomixis field, and so Apomixis research tends to go in kind of in bouts and waves, essentially. So back in 2001 we had the meeting in Como and excitement at that, and then there's periods of lulls, and then it picks up again. And right around now, and accelerated by, I guess, the ongoing advancements in genomics and the toolboxes such as CRISPR and whatever, and long-read sequencing that we have, it's clear that there are a number of groups that are converging towards making Apomixis prototypes, and that's really exciting. And for the first time, in my mind at least, it does herald the possibility of apomictic crops engineered by one means or another to actually make it into farmers' fields.
Okay. The other Charlie, what is your feeling about the presentations and about the status of the field?
Yeah, I mean, something that was very nice that came across very clearly to me today is the kind of two different approaches that are being followed in Apomixis research. Is the kind of synthetic approach. So, for instance, how Kejian has shown this modifying meiosis into mitosis and adding in haploid induction approaches. And then you have also the work of Tim and also our work on Taraxacum, which is really looking in natural apomix and trying to harness how natural apomix have modified plant reproduction. So I can see that in the next years, the kind of combination of these two approaches will probably lead to even better prototypes of apomixis in crops.
Eric, what would be your view or your reaction?
Well, I enjoyed very much the different topics that were covered, and I think all the speakers gave us a nice overview of their particular systems, but also reflecting on the general mechanisms. And I think that's maybe, I agree a lot with what Charlie said, and both Charlies had to say as well. But I think what I like about thinking about the different systems is it also helps highlight the complexity, which also was the kind of beginning of our talk too, that sex is complicated enough. Asexuality is probably even more complicated than sex in some ways. And I think that's what's cool, is you get a feeling a little bit for the commonalities of potentially, for example, male function being taken over by the female, for example, across multiple systems, or changing gene function, like how important that is. But what gene's being changed, or where that gene is in the genome, those are where some of the small flavors come up of the different systems and the complexities, right? So it's this kind of balance between what are the rules that might unite Apomixis, various apomictic systems, and what are the peculiarities of each system that make it complicated and fascinating to hear everybody tell their own different story and their own different approach. And so how can we learn from Buchra, or how can we learn from Taraxacum, or what can we learn from monocot systems where we're doing that? Like what do we need to figure out to make it work in no matter what crop we want to work on? And I think that's one of the exciting things and also the challenges for the future.
Okay. Peter?
Yeah,
Live from Wageningen, what is your idea?
I agree with Eric that I really enjoyed the diversity in the three talks. gives a good flavor of what's possible. Also, I think, but that also has been said before, that it clearly reflects the new developments in the technologies. So we have these long-read sequences. So for the first time, I think now we can fully sequence the Apomixis loci, and we can see what is in there. So before that, we often had gaps, and when you have gaps, you don't know what is in the gaps. Turned out often that it was not very important what was in the gaps, but anyhow, it's good. And then also the CRISPR-Cas technology, so we can really target all the genes. Yeah, I think that really is the technological developments are very important.
So what else would you need to make a breakthrough? I mean, we had the sequencing technologies indeed, long read, CRISPR-Cas to make quick mutants, even in the difficult plants like Taraxacum or rice. What would be your dream about technologies which really bring us further, which gives us an insight what happens in this hidden female reproduction part?
Yeah, maybe we should go more into what's really happening in the fundamental cells, so in the egg cells, cell-specific transcriptomics. Although I'm not completely sure whether that's really the most important way to go. I still myself feel that good genetic mapping combined with sequencing and expression studies can be very useful. And especially with these new developments, what I just mentioned, I think we still can use these and see how we can go further.
Do you think you have all the tools already? Or
yeah.
Do the others agree? The others
may I just add on?
Of course, of course.
So I think at the moment the long read approaches are very powerful, and they are pretty universal in their application on plant species. If you can get good high molecular weight DNA, then you can sequence the genome of pretty much any plant. But the missing element in some of the wild Apomix will be how we can do genetics or how we can mutate the genes. So ways that we can deliver CRISPR-Cas universally would really help, because I think being able to do genetics on those species is really important.
I see Charlie also nodding his head.
Yeah, like the point of the transformation bottleneck to, if we want to be able to use the CRISPR-Cas or genome editing new capabilities, particularly in apomictic species, to understand function, then we need tractable transformation systems, which is Charlie's point, and it's very valid and well made. Like the advances that we see have arisen from, pushing on sexual understanding of sexual reproduction on the one hand, and trying to decipher components of apomixis from sexual reproduction, while pursuing the even harder making progress on the natural apomix themselves. And now we're seeing a really nice convergence whereby we have a toolbox that is derived from both of those efforts. But I think to put it into practice, particularly in many crops, we have a transformation bottleneck that still exists.
And what is the panel's vision on the amount of genes which you maybe need to start influencing, or that you need to introduce apomixis in whatever way, natural, crossing, or synthetically? What is your opinion about that? I have an impression that Rik here says maybe 1 or 2. Tim says 20. Maybe more.
So if I just might, so once again, if we think about apomyosis, parthenogenesis, and endosperm formation, right? So, and then of course you'll have your handful of genes which seem to be associated with those things. But once again, think about it. I think the things we need to be thinking about are sex chromosome evolution, heteromorphic sex chromosomes, and how that process goes from autosomal sex determination, single locus, and then kind of divergence. We need to think about that. And we also need to think about cytoplasmic factors. And for example, the absence of selection on male alleles as you go through the female line, and mutations which accumulate in cytoplasmic factors. I think, so what I can tell you is in the genes that we have discovered, we have all of these things. And as I said, there are some factors, for example, which very clearly shift expression from male tissues to female tissues, right? And so it's so it's not only the genes per se that switch that trait on, but it's the genes associated with how those genes got segregated into for example, these hemizygous regions. So I'll stop there. That's just simply what I wanted to say when we're talking about these handfuls of genes.
But you don't believe in an overarching regulator, then, Tim? Which
I'd love to. If I hit it, yes, you'll all see my nature. Yes, I mean, absolutely, right? But I think it's not as simple an evolutionary trajectory as we may consider, right? Like there's, it's a multigenic trait, and we've always said, okay, well, it's hard to kind of imagine how those traits come together. If you have apomiosis without parthenogenesis, right, then you end up with ploidy increases and so on, right? So and the other thing, I guess, is you do end up with these hemizygous regions, right? So like the hiding places for all of these factors, as we see in Y chromosomes, right? And then, yes, it does bring up the idea that maybe there is kind of a universal factor which kind of influences them all. Definitely, I didn't show any of this, but we do have a handful of factors we've identified as well, right? So yes, I'm hoping that comes out as well. So
Peter and Erik, what do you think? Peter?
Well, yeah, if I may, so Tim is raising these regions of non-recombination and combining it with sex chromosomes. But I wonder whether these characteristics of these loci are not secondary effects of long-term asexual reproduction. So in there somewhere there will be a ruby in the rubbish, some people call it. So if you can find a ruby, then and we leave the rubbish out and just introduce a ruby in the sexual plant species, I think. Yeah, so that's my thought about that.
Charlie Spillane, what do you think? Not a magic bullet, probably, but
no, but we're at a very, or like the teams here are at a really good point, whereby incremental, and perhaps seismic kind of shifts can be made in terms of, particularly like we take the case of penetrance for parthenogenesis, like so currently at about 30% as the max, but like that's a great place to be because there's a lot of potential for tinkering around with modifiers and promoters to try and drive that higher. And because at some point the penetrance has to go up for it to be a usable system. So it's brilliant to have the prototypes to tinker with, and the tools are there to do that. So I think the vista looks very promising.
And Charlie Underwood, what is your view on how to introduce Apomixis before we go to the more applied aspects?
Well, I mean, I think in terms of the gene numbers, I think from the work of Kejian and from also Sundaresan's work in UC Davis, then we know that modifying 4 genes can be enough to get clonal seeds. Another thing that springs to mind is actually mainly we're talking about gametophytic apomixis, but you also do have this sporophytic apomixis where you basically get a embryo developing from the sporophyte directly, and the work in citrus suggests that might be just a single gene. So maybe it's as little as 1 gene might be enough. But I think that there is still probably work to do, as Charlie just mentioned, about improving the parthenogenesis, because I think that is the limiting factor at the moment, and getting that to as close as 100% is what's required.
Okay, so let's go to we have still 6 minutes left, I think. Let's go to maybe the application. Of course, there is tremendous, let's say, room for applications also. Clonally propagated seeds. It's the golden bullet of, some people say, of plant breeding. Do you agree with that? Especially if you let's say compare it with the loss of hybrid seeds, which are now giving a lot of quality improvements. What type of crops would you like to apply this technology first? We heard Kejian in rice, of course, but are there any other crops which we should start working on Apomixis? Who wants to say something about that? Tim.
Pulses. So I think we need to think about selfing crops, right? So cleistogamous crops, which are highly homozygous, right? And we don't utilize hybrid breeding strategies. And so I certainly see that as one big group that needs to be worked with. Okay.
And what does the panel think about, this is going to be a clonally propagated crop, a monoculture. There's a question from the audience. Laurie, what about plant disease problems in clonally propagated crops? Is there a big risk on that?
Can I just comment on that? So for propagation through seeds is actually quite useful for removing viral and pathogen loads. And so, you know, Apomixis, if we can manage to move it into clonally propagated, vegetatively propagated crops such as potato and cassava, yams and so forth, could be of major benefit for phytosanitary reasons to have better planting materials. So in some sense, if we look at about 10% of the world's potato crop is used for 10% of the world's potatoes are used for planting, whereas if we had true potato seed as enabled by Apomixis, then that would be a major benefit, I think, in the clonally propagated crops, vegetatively propagated crops.
And Mr. Wang, what are you thinking about that? What do you think will be the risk of diseases in your clonally propagated rice populations? I don't know if he can hear us. He seems to be... Professor Wang? Can you hear us? No? Okay, let's continue with the other one. Do you think we can have other applications of the genes involved? We are always thinking about Apomixis, but we also have the PAR gene and the let's say the fixation or the prevention of meiosis. Can we think of any other applications? Peter, maybe?
Yeah, well, something very obvious would be unreduced gametes for polyploidization, transmission of a lot of heterozygosity. For instance, the DIP gene in Taraxacum has no recombination and is unreduced, so that would be ideal to basically you transfer 100% of the heterozygosity. Yeah, I know in the past, that's already some 15 years ago, that longer ago even, that there was this idea in potato to make apomictic potato, and there also people tried to get together the individual component of Apomixis to make apomictic potato. And there it was very important to have the good unreduced gametes, so the ones that do not have the recombination and have the good transmission of heterozygosity. So that would be one application. Of course, also haploids, dihaploids going up with ploidy level, but also going down to ploidy level, connecting different gene pools, so a tetraploid gene pool with a diploid gene pool, for instance. That can, yeah.
Okay. Kejian, I heard that your microphone is on now. Would you like to react on the risk of diseases in your clonally produced seeds derived, the crops derived from clonally propagated crops? Can you comment on that? What is the risk?
Till now, we didn't find any difference from the wild type. But you talk about risk. Because it's a colonial plant, and many people talk about it maybe like the banana, maybe in the same plant growing in the fields, that it will increase the risk of disease or pests. But I think if we synthetic apomixis is widely introduced, it can be introduced into different varieties. So I don't think the genetic diversity will be done. I think the we will increase the genetic diversity in fields, rather decrease the diversity.
Okay. I think we're coming a little bit to the end of the workshop. I have a question to. I see that Rik has let's say connected again. I have a question to Lucia and Rik, and probably also to Rik. How far would you estimate we are away from an apomictic, let's say, lettuce or rice or sunflower? How long will it take in your, if you listen to all the presentations and if you assess the point we are, how far are we from such a potential apomictic crop? Lucia, what do you think? Is it months, years, decades? You're on mute, Lucia.
Okay. For sure, not months. I would say years, or yeah, quite some years. I would say yes. And
what do you need? I mean, do we need more understanding, or do we need the tools, or?
But the tools, as was already mentioned from many of our colleagues, they are available. So except for the fact that if you want to introduce to crops, of course, you need also to be able to first understand in very much detail the genetic network controlling apomixis. And this required, as also was mentioned before, a good functional genomics. So we really need to create mutants and to study the genetics, the genetic base of apomixis. So in terms of tools, I think we have already discussed with the committee that there is still some need in develop methodology to be able to indeed create mutants and the CRISPR-Cas9 line. Of course, this will take times, and but this is, I think, is needed to really be able to develop a strategy. So first, to really understand, if we start from apomitic plant, to really to understand the genetic basis, if we want then to engineer. Another type of approach, as was shown before, is to start from sexual reproduction and try to engineer sexual reproduction to make it asexual. So depend a little bit the strategy that want to be chosen to see which type of technology are still needed to develop this strategy. But still, I guess some years at least.
Okay, let's make that a conclusion. And I would like to thank all the speakers, all the members of the panels, for your very much appreciated contributions, also for the discussion. I would like to thank also the technical people who is here. I would like to thank the audience for being here and attending. We had more than 450 attendants. When you would like to learn more about KeyGene, please visit our website. Would like to also invite you not only to the sexual plant reproduction in Prague this summer, but also to the crop innovation and business event, this time in Ghent, Belgium, 27th, 29th of March. During this event, we will gather with more than 150 research managers and scientists, CEOs, to discuss potential developments and influential developments in the technology innovation for crop improvement area. We hope to see you there. And I would like to thank you once again. My name is Arjen van Tunen. I'm the CEO of KeyGene. And thank you for your attention. And see you next time. Bye-bye.